nice to be back in San Diego after almost 4 years, and then meet lots of old friends, make new friends. So today I'm gonna share with you guys what have we done in our company using stem cell to do drug discovery. Actually, for disclaimer, we don't do cell therapy. We use this as a platform to develop new drugs. So, but the core technology we have is stem cell based. Can I drive from here? No. And next slide. Yeah. So this probably is something most of you are familiar with. The challenge of developing new drugs for neuroscience is really the high failure rate. If we compare all the indication, you found one in the white one, that is the lowest success rate. That's the thing as well. And then if you look at where the failed, you will find one thing interesting is it's the most in phase 1, in phase 2. And then when you go to phase 3, actually the success rate for neuroscience is not that much worse compared to other indication. So that's tell you one thing, that is when you decide to move this compound in a clinic, right? Right now we mostly based on the animal model. The preclinical data are really not a good prediction of what happened in clinic, right? So in phase 1 failed, that means you have really a high false negative rate for the safety. And then if your phase 2 failed, that means you really have high positive or high false positive rate for the efficacy. So that is the biggest challenge of the drug discovery neuroscience. Basically, it's a loss in translation, a translation from clinical preclinical to clinical study. So that's our problem. And then you look further, why the fail in terms of accuracy. So this is a summary of all the reasons for failed. If you look at the biggest failure, 40%, is probably the missed link between the target and the disease. Most are the target based, but the missing link actually becomes between target and the disease. For example, we work on the AD and Tau, but whether those are the driver of AD, I think still debatable. And then most important is we don't really have not very good validated models for new disease. So that's the problem created. So why is that? Is as mentioned, we mostly use a mouse or rat model to test the efficacy and safety of the drugs. And then if you look at the brain, the mouse brain and the human brain is very different. Evolution is 75 million years away. And then look at the structure, human brain has all the curvature, and the mouse brain is flat, right? And then so, there's a joke is, we are very good at making the mouse sick, right? But we can't really, never kill them very well, but, it doesn't work in human. The reason is, is the mouse is very different from human, especially in the brain. That's the fundamental difference. So no matter how good you make a model in mouse, it's just not human. So the problem is because mouse really hard. It's hard to use mouse to really recapitulate human biology. So that's the case. So what we need is we need to do human biology. And another thing, if you want to understand, how to increase the success rate of discovery, look at the histories. So when you go to a meeting right now, even for neuroscience, most people are focused on target-based discovery, right? This has become really successful, mostly actually in the oncology and the autoimmune space. But if you look at back to the success of neuroscience, it's different, right? Because if you look at the history, origin of the first drug ever discovered is the penicillin. It's based on phenotype, right? So it's phenotype screening. So this is another interesting study. You look at, this is some between 20 years of success of all drugs in different space. It's NCE, either a small molecule or biological, and it tells you how the compound was discovered. You found for the new class of drug, mostly are based on phenotypic screening, no matter what indication. There's a high that has the highest success rate. Especially you look at the CNS. Of the 8 drugs that become the new class drugs, only 1 is target-based screening. Most is from phenotypic screening. And then this is actually common now in neuroscience. Even with a lot of drug we use in clinic all the time, we call them a dirty drug because we don't know what target they are. Even if we know target, it's most likely more than one target. So that's two things we understand now. One is you don't really have good human biology in drug discovery, and secondly, target-driven discovery for neuroscience particularly is very troublesome. So we need to think about how to revise our way of thinking how to develop new drugs. So the solution is one solution is use in human stem cell. So this is probably very familiar for you guys. This shown in many reviews, similar graph. So the idea is we take human cell, right? This is pretty true for the genetic disease because a lot of new disease is genetic caused. We take a human cell, either from the skin or blood, with Yamanaka factor, turn them into stem cell, right? And then the stem cell we can program to the cell type you want, being the muscle, be neuron, and then this is the model. And then because this cell came from the patient, it has the same genetic background of the patient. So whatever the phenotype you see in these cells, they should be mimic or recapitulate what the pathology in the clinic. That's a phenotype, So with that, you can do drug screening, you can do drug validation, and then go back to the patient. So this is the paradigm to use this. And then this gave another way to make a cell to drug discovery. And this particularly interesting is for neuroscience because, for example, oncology, even though we have great success in past 10, 20 years, one major reason is the PDX, right? You can take tumor cell from patient, put it in the mouse and carry it, right? That's, kind of, reserve the structure and the biology of, the tumor. But you can't do that with human neuron because first one, it's hard to get the human neuron. That's very difficult. And secondly, neurons don't proliferate in vitro. You can't grow them. So that become a big issue. And then stem cell makes that possible. So as mentioned, basically the company we started almost 8 years ago is first on building this, we call it SynFire stem cell platform. Syn is synchronized. And then fire mean in neuroscience, we also say the network bursting is the firing. So that is where it come from. So the idea is we take a donor cell, right, from either a healthy subject or a patient. We take the somatic cells, either be blood or in the US, more common is the skin, whereas you have a fibroblast. And with that, you turn to the iPSC, and then you can use multi-way to program them into the cell type you want, it be neuron or glial cell or microglia. And then with those cells, you can put together into this called a co-culture system, and then the co-culture how well-defined what you have. And then, or you can do, become more trendy now, you can do the 3D co-culture called organoid, or you can transplant into the mouse called a xenograft. This is particularly interesting for some cell like microglia. It's hard to mature them in vitro. So it's easier to put into mouse to the xenograft. So this is pretty common, and this has been tested for almost a decade now. It is how things work. So what the particular about our platform is this. So we use this technology called the reprogramming. So this is different from most methods. Most farmers is you have a stem cell, you use the TO culture, use all kind of growth factor to guide the differentiation. That's kind of mimic the natural development process. So the good thing is more follow the human biology, but the problem is it takes a really long time. Sometimes you have to go through the stem cell stage, the progenitor cell, and then a couple steps, and then in the end, the population may not be what you want. So what unique about our platform is we use a particular combination of transfer factors, use virus to introduce into the cell, basically to force the cell into a particular pathway of differentiation. So with different combination, we can control the fate of the cell. For example, with one factor, NGN2, we can program the iPSC into glut neuron. Basically, it's an excitatory neuron. And then with the different factor, we can program into the GABA neuron, the inhibitory, or into motor neuron. So with this way, you can really fast, within months to make a very pure population of the cells of your defined genetic background and pure. And then we can, cryopreserve them. So when you do experiment, you take note, use whatever combination you want, the ratio put together, do co-culture. And those cells really show a very typical linear cell here with also biomarker to show it's actually the cell type we want. And we also did a lot of calculation, especially in neuroscience for to understand neuroscience is to use electrophysiology. So they have all the receptors, all the ion channels and all the network activity. So they can form premature system in vitro, like in 3 to 4 weeks. Then you can do experiment on them. And then we also, in the process of working to make iPS-derived glia cells, because in the human brain, the most important immune system is the innate immune. The major 2 component is astrocyte and microglia. And as you probably know, almost all the disease in the end is an inflammatory disease, especially for neurodegenerative disease. So it's very important to be able to model this immune response aspect of any disease. So we can make astrocyte, make microglia, and then this is some data generate. You can look at the RNA-seq profile. It's mimic, the human astrocytes, and then you can do a lot of function assay. So actually, they respond to all kinds of stimulation to secrete the cytokine. So this is make all the subtype, individually and then combine. So when combine them, we can make into 2D. So this is very pretty one. So this movie show from stem cell, while program them, it start to grow this neurocyte, very long neurocyte. So within 7 days, they're gonna form this very beautiful neural network. So this is time-lapse in 7 days. It's basically have all the network. And on top here is the mature network you can see. And you can use different marker to label different subtypes. So this here basically you have 3 types. With label here is inhibitor neuron, the essential neuron, and actually it's also have astrocyte. So this is 3 component form the basic neural network, which is really important. And then you can grow this in a 2D format, or you can do it grow in the 3D format, it's organoid. And with that, you can do drug discovery, you can do drug validation. So the advantage of this platform, first, is real human biology. You use a human cell to look at them. And then you can really model disease relevant interaction. For example, we can model neural network. We can model study, for example, EI imbalance. It's very important for neuroscience. And then with all the immune cells, we can also model the immune aspect of it. And then because this cell we produce separately, so it's a very modular system. So we can control the genetic background of each individual cell type. That's very important when you try to model disease. Also, this is a pretty fast process. For example, the typical production size section is a couple months. So we can develop this model like within a year from beginning to have assay ready. So we also can do it in 3D. We call this assembloid. This is a little different from most people do in organoid. So in organoid, you have a stem cell, you grow them into the organoid, but that problem is it's very mixed population. You really can't control them. That's, I think, one of the reasons that even organoids has been developed for many years, but it's not be able to use for drug screening because it's not scalable. There's low reproducibility because the nature process can't control. So what we developed is making the individual cell type and mix together with the control the ratio and control the growth. So in vitro, you can form this very nice ball of cells, and you can look at them. They have all cell type there. So this is called assembloid. This can give you more control the process, so we can really scale up this process to do the discovery. And also, there's recently a paper published by a collaborator, Yulin's lab from UCSF, showing, for example, this is the Parkinson model, TB43. If you use a 2D model, same cell, you go to 2D, you can't see the phenotype. But if we go to 3D in assembloid, you can see the phenotype. So that's how important, the spatial structure is important for this in vitro model. So that's why we always try to not only look at the 2D, also 3D. Also, actually, we found out for certain phenotype in 3D, it's much faster. For some phenotype, if you're 2D, you probably take 3 months to see the phenotype, but in 2D, you probably can see in a month. So that's very interesting. So that is the biology part. But again, to develop a screening platform, you also need a hardware part. So this is some work we have done, some in progress about the building this called microphysiology system. So basically, you model not only single cells, you want to put together to mimic more the physiological condition of human tissue. Have all the components, not just a couple cells. You also model the cell-cell interaction and also all the matrix in it. For example, Emulate, a company in Boston, had developed BBB with our neurons in it, can model the blood-brain barrier pretty well. It has all the cell types there. And also we are in collaboration with some company look at the neural growth in the microfluidic devices. You can measure not only the neuron itself, you can measure the size, the length, the speed growth of neurons. And then we're also working with another company, is another startup in Silicon Valley, to try to develop a high throughput system to combine with our system to enable a really high throughput screening. So this platform can enable, we not only can do the small scale lab stuff, but we can become an industry scale screening for high throughput screening. And the screening is using not only a simple cell system, we're going to have the MPS, the microphysiology system, to do the screening. So for the screening readout, now we have a couple we're working on. So one is for the 2D or 3D, we can do the middle throughput, high throughput screening, for example, with electrophysiology. I think some are familiar with this platform. This is called a microelectroarray system. It's a system where you have the electrode at the bottom of the tissue culture well, and the neurons go on top of that. And then you can record the change of active potential, which is basically reflect the natural activity, network activity of the system, not just individual cells. So that's very important because that only can tell you whether the system is mature enough, and also whether you can really capture something that's really subtle, but only on the circuitry and network level. So that's one. And this one, we can do like a media throughput screening, like a couple hundred compounds a month. And then with a high content imaging, you can really go high throughput. This is a paper published recently by Bing Qi, one here still in Genentech, to show you can do fully automation and then use the neural groups and then some phenotype to do hydrogen screening. So this is the system that we and other player in the industry is working on right now. And then, so that is the model. The next question is, this is particular for the brain is, when you have a drug, can you really get into the brain, right? Brain, we have this BBB, is very tight control system, environment for brain. And then the BBB here show you have, all the cell types, not only the parasite, the endothelial cell, but you have, for example, astrocyte is playing a big important role. So can you really model the BBB in vitro to make sure whatever drug you're making can into it? So that's something we're working on. And then there's a model already working on that. And then the next question is, when the cell can get into BBB, can the drug get into the cell you want? Because a lot of gene expression is very cell type specific, right? Where you got a neuron, you got a microglia, you got other side. And then the co-culture system we are developing because they're well defined. We know exactly what is happening, so we can test the cell type specific gene delivery. For example, for gene therapy and the expression of the payload in cell type. This is some work we did for the collaborator to show you can use the in vitro system to really test, for example, the efficiency of different AAV to get the neuron and which one is best. And then you can see the very different. For example, this particular cell type is the most efficient one. And then after that, you can look at the expression of the gene you want to deliver in very cell-specific function. Exactly in which cell is going to be expressed. and which in which can cell the gene can be knocked down. So this is can ensure the drug not only can get in the brain, also get into the right destiny of the drug. So the next question is, the drug work, and then whether the drug can safe. And this is actually another big challenge for neuroscience drug discovery. Again, this is a review published almost 10 years ago, saying say what happened to the drug when you test them. So here the right bar is that the drug failed in preclinical, right? And then the yellowish brownish color is the number of drug failed in the clinic. So when I look at this, one thing you can strike out, CNS very different from other profile, right? Most drug in the CNS area failed in the clinic. It's very dramatic. Why? Because most model we use to test the toxicity of any drug, not only the neuroscience drug, in the predicate is very unpredictable. It's pretty easy to understand. For example, when you look at a drug into the mouse could be sleepy, right? How you can measure that? But in human, that's a bigger problem, right? Because the model right now, you can't really measure a lot of subtle side effect of the drug, especially for the brain, because brain is very sensitive to a lot of stimulation. So one model we developed first when we started a company 7-8 years ago is look at the neurotox. Can we use this very simple system to look at whether the drug is safe or not? And then for the neurotox, the biggest tox liability is seizurogenic. So basically, the compound gonna disrupt the EI balance in the brain to induce seizure. So that's one talk. And then the neuron become too active, that's a seizure, right? If it become less active, it have a sedation effect, making you more sleepy. So this is a good starting point for us. So what we did is very simple model here. So we have this co-culture system with a healthy subject developed neurons, the wild type. We put the co-cultures together in 3-4 weeks. They form very mature neural network. And if you measure with MEA, they're gonna show this called synchronized network bursting, very arrhythmic at a certain frequency. And this is very similar to human EEG. This is basically the brainwave, right? And then if you put a drug onto the model, it's gonna change the different pattern of the system. For example, here, you become hyperactive. You see this long, prolonged bursting. And that's the seizure. And that's what exactly you see in a patient when the seizure happened. So this is a model for drug testing the neurotoxicity. And then the question is, this is very simple cell-based. A lot of people, especially people familiar with the animal models, this is too simple to be true, right? Then in biotech we have a saying that data, right? Let's look at the data, what happened. So we did a lot of data. So we test about 50 compounds. Different compound has a neurotox problem, and then see how good they are correlated. So here is this small panel here. It's one of the panel we work with EPA that's published a couple of years ago. This is our first panel. We test this compound. This is most of the called environmental tox. This is a pesticide. The EPA very concerned, whether the chemical can have, effect on human brain. So they test this one. And then the gold standard now is, red cortical neuron. So we did this work with EPA to show our, model is, equal better than the red neuron. And then they publish. And then we later test, additional panels. They have give really good, prediction whether this our system is accurate or not. It's have 90 to 100% accuracy quantitatively, and we can also do semi-quantitative analysis. So, and also beside this, interesting is we can not only to say this drug work, we can also do targeted convolution. So, for example, those drugs we know they are two class target, and then we train a neural network simply to tell us which class of target is hit. So it gives 80% accuracy. So this is pretty amazing. So again, this is how we think about change the paradigm of doing drug discovery. So we're going to introduce the human biology early on in the drug discovery process. So as the early screening and observation stage, you use a human cell to do this, and then you can follow the flow to do the rest. So time out, right? Okay. Yeah. So there's a bunch of data I will show, but I think that's for the same time. I only want to show one data. This is pretty interesting. Here. This one model I'm most proud of. So this is epilepsy model. So what we do is, as part of continuum or tox model, you induce seizure, right? And then you put an anti-seizure drug on it. For example, here is lamotrigine. And then you can see a dose response curve to the drug. So you induce seizure, you reverse dose, and the dose dependent matter. Okay, so this is one drug. So this is, the 18 drugs have been used in the US on the market. So the y-axis is saying, this is drug, what dose going to be the drug when patient taken them. So this is efficacy dose of drug in clinical data. And the axis is the EC50 we measure in our system. So you can see pretty much linear regression. So remember, this is not the binding assay. This is not the chemical assay. This is the functional readout. So this will tell you how good this model can be. So let's zoom through. I think this is probably the last slide I want to say, because last December, there's a major change in the US. So now FDA don't have the mandate use animal model for efficacy. It's optional. So this is a major change in the regulatory regime in the US. This kind of open up a lot of interest in doing cell-based assay. But also this is nothing new. For example, this is drug, this is a Sanofi drug for a rare disease. They got approved, I think 2 years ago, purely based on the efficacy data from in vitro model. There's no animal efficacy about it. So this is the first time ever that FDA approved NDA filing based on in vitro model. So I think just the FDA is open now to use in vitro model for support NDA filing, as long as you convince them the data is good enough. I think my time off. Okay, thank you. Yeah.